Good morning, all. I'm M. Kezia Elizabeth, a lecturer in microbiology from TSWRTC, Warangal East. Now, as of now, we all are familiar with the term virus, isn't it? Because of COVID-19, how the entire planet got locked down, and we are passing through the different phases of unlock. So we all know what exactly are viruses. So that's my topic for today. We will discuss about the general characteristics of virus. The term virus is a Latin word which means poison or venom. So in 1886, Adolf Meyer was the first person who observed viruses in tobacco mosaic plant, and this virus was called as TMV virus, that is tobacco mosaic virus. Virus is an ultra microscopic structure which is very infectious, and they are metabolically very inactive and will multiply only within the living cell or host. Now, coming to the general characteristics of viruses are metabolically inactive, so they are considered as obligate intracellular parasites. Viruses cannot make energy or proteins independently of the host cell, so viruses multiply inside the living cells using the host machinery. Hence, therefore, we call them as the obligate intracellular parasites. Viruses were considered as living things because of the following reasons. That is, they occur in different strains, they have their own genetic material, and they can undergo mutation, and they can be destroyed either by ultraviolet rays. Now, there are some non-living characteristics of viruses as well, like they lack a cellular structure, they do not respire, and they do not move or grow, and they require a host cell to move or to grow, and they can be crystallized and stored in bottles like chemicals, and they lack enzyme system, and they do not have a metabolic activity of their own. Habitat. Now, viruses are able to survive in different type of habitats, sometimes with the help of a host cell or sometimes with the help of any other source. So active viruses mostly, which are very pathogenic, require a host cell for multiplication. They are present either in the bacterial cell, animal cell, or plant cells. So the examples of different type of viruses like herpes virus, TMV virus, bacteriophage viruses are the different viruses which are present either in the bacterial cell, plant cell, or the animal cell. But we have a set of viruses which are able to even survive in human cells as of now. Now, coming to the sizes of viruses are smaller than bacteria, and they can range in size from 20 to 1400 ml. Like we have a polio virus which is 30 nanometer in size, or we have a giant mimi virus which can range up to 800 ml. Now, we have got different shapes of viruses. So these shapes of viruses can be either in rod shaped, bullet shaped, filamentous shaped, icosahedral shape, or tadpole shape. You can see the images of different virus like the measles virus, bacteriophage virus, and the rabies virus, which come under this various different shapes of viruses that can occur. Now, coming to viral structure, viruses consist of nucleic acids and proteins, and the genome of the nucleic acid is covered by a protein coat. So this is called as a capsid. So some viruses which have got an envelope outside the capsid, so we call them as the enveloped viruses, and some viruses do not contain any capsid. Outside the capsid there is no envelope present, so we call them as the naked virus or non-enveloped viruses. So this is the typical structure of a virus that you can see in the image. It has got a nucleic acid which is present inside a viral coat, and then there is a capsid that is surrounding, which is again covered by an envelope. So if there is an envelope present, we call it as an enveloped virus. If there is no envelope present, we call it as a non-enveloped virus. And the nucleic acid is surrounded by a capsid, and the capsid is usually proteinaceous in nature. Now, viral genome. So, the genome of a virus can be either DNA or RNA. So, deoxyribonucleic acid or ribonucleic acid. So, depending on whether it is containing DNA as the genetic material or RNA as the genetic material, we call the viruses as DNA viruses or RNA viruses. And can be single stranded, that is, the genetic material can be either single stranded or double stranded. Then they can be in different shapes, like the DNA can be circular in shape or linear in shape, or the RNA can be circular in shape or linear in shape. Now, coming to viral capsid, viral nucleic acids are surrounded by protein coat. So, that protein coat is usually referred as a capsid. So, depending on the shape of the capsid, there are three different type of viruses. That is, we have helical capsid, we have icosahedral capsid, then we have a complex structure of a virus that is called as a complex capsid. So, helical symmetry. You can see the image of a helical virus. Here it is a filamentous capsid. It has got a long tube of protein with a genome which is present inside that. Now, the best known example of this is influenza virus and the rabies virus. So, we have the protein subunits, and these protein subunits are getting arranged in a helical symmetry. So, you can see the image of a helical capsid virus like the rabies virus or the influenza virus. Similarly, we have a cubic virus symmetry, which is also called as the icosahedral symmetry. So, it is made up of icosahedral capsid. There are around 20 triangular sites. The example of icosahedral symmetry viruses are HIV virus, hepatitis B virus, or the adenovirus. Similarly, hepatitis B virus structure that you can see is a icosahedral virus, and you have structure of HIV virus which can be seen in the diagram, that also comes under a icosahedral virus. Okay, now coming to the replication of viruses. Since viruses are obligate intracellular parasites, they require a live host for multiplying and replicating itself. So, the viral replication is basically classified into two types, that is, lytic cycle or lysogenic cycle. Now, what is a lytic cycle is? It is divided into 5 steps, that is, the virus needs to first attach itself to the host cell, that is referred as the attachment. Then it is going to penetrate inside the host cell, which we call it as penetration. Then once it penetrates inside the host cell, it is going to release its genome inside, and then the replication of the nucleic acid of virus occurs using the host machinery. Then once the replication is done, the maturation of the virus occurs, where the assembly of the virus into different viral capsids occurs. Then the release of the newly formed new virions occur by the lysis of the host cell. Since the lysis of the host cell occurs and the virus particles are released outside, we call it as the lytic cycle. Now, in lysogenic cycle, we see that the virus enters inside the host cell, it integrates inside the host cell, inside the host genome, and whenever the virus is replicating along with the host genome, the replication of the viral genome occurs. So, we call it as lysogenic cycle, and these lysogenic viruses are getting transferred to the daughter cell whenever the host cell gets replicated. So, these are certain general characteristics of viruses. So, let's have a summary of what all we have learned for today. We saw the different types of viruses based on the shapes. We can call them as the icosahedral, or we can call them as the helical symmetry, or cubic or complex viruses. We saw certain general characteristics of viruses based on the genome that is present, whether they are RNA viruses, DNA viruses, or they are enveloped viruses or non-enveloped viruses, and we saw the different life cycles of viral replication. Thank you all.